Mushrooms inspire fear and fascination. We see one and we wonder, can we eat this? Will it kill me? They take forms unlike anything else in nature, visible structures of fungi we can't easily see. These fungi do so much more in our ecosystems than we give them credit for. This is Coast to Canopy, an ecology podcast from WFSU Public Media. I'm Rob Diaz de Villegas. With me today is Matthew Smith, professor in the University of Florida's Department of Plant Pathology. Welcome to Coast to Canopy.
Thank you. Thanks for having me.
And also joining us is his PhD student, Alassane Sow. Welcome.
Thank you very much.
All right, so let's start with earlier today. You guys drove in from Gainesville to here in Tallahassee. We went to Wakulla Springs State Park. So describe sort of what we did and how far we trekked to find all the mushrooms that you have right there.
Not very far. So we spent about 2 hours in the woods, more or less. And I would say we went between 100 and 150 feet, probably. Not very far. It wasn't a long way. And we found quite a lot of fungi.
Wakulla is a nice hardwood forest. And because of the rain, it's been wet. And because it's shady, it stayed wet. And because of that, a lot of mushrooms have come up from wood, from the ground, and on trees and all kinds of things.
We see here, and I've taken photos that we'll be editing in so people watching on the video version will see this. And if you're listening, you might want to check it out or go to the blog, wfsu.org/ecology blog, see a lot of the mushrooms we discovered. So a really neat diversity of mushrooms. And talk about one thing you said this morning that kind of struck me is sort of the estimated number of fungi that might be out there versus how many we actually know.
Yes, it's a pretty big difference. So we know, especially I would say in the modern era where we've used DNA evidence, we know the diversity of fungi is really high. So the estimates fall somewhere between 2.5 and, 4 or 5 million, depending on who you believe, in which study you listen to. But it's a lot of species. But there's only about 150,000 species right now that have a Latin name and are formally recognized. So this huge gap between what we know is out there and what is sort of has a Latin name. In comparison, plants, there's a little over 300,000 species. And so, in comparison, there's a lot of fungi that we still don't know what to call them, and they haven't been officially discovered by science, but we know they're out there.
How likely are we to see something that's undescribed by science?
You'd be surprised, actually. Yeah. So an example would be this Parvodontia that we described, and one of the members of our lab, Claudia Paez, she described the fungus along with collaborators, and I mean that's a fungus that we know has been in Florida for a while, but. You have to be looking in the right place at the right time to find these fungi. And then you have to determine that it's new, which is also challenging for some of them.
And so you talked about DNA sequencing. And so we have, even here within here, you pointed out the coral mushrooms that, they look alike, but then a lot of things look alike that evolved in different. So talk about this aspect of it that a lot of things you see sort of their evolutionary paths, I guess.
Sure. Well, I guess almost any ecology that you see in fungi and almost any morphology that you see in fungi has evolved multiple times. So that's a common theme among fungi. So for example, I study a lot ectomycorrhizal fungi. These are the fungi that are symbiotic with the roots of trees like pines and oaks. And this is something we know has evolved more than 70 times in different groups of fungi. So the fundamental way that they get their energy through this symbiotic association with plants that shows up a bunch of different times in different groups. Similarly, we looked at coral fungi and we saw 3 different ones. 2 of them were mycorrhizal ones and one was a saprotrophic one. And those, they look quite similar. And if you didn't know already what they were, you'd say, oh, those are a bunch of coral fungi. They're probably related to each other, but they're in 3 different orders. So that's an example of convergent evolution. And certainly convergent evolution happens in a lot of groups of organisms. But in fungi, I think the soft fruiting bodies, they can change really fast in evolutionary time. It makes it hard to study fungi because when you look at them, you can't always tell right away which group you're dealing, which order they belong to. So that makes it a challenge.
You were looking at things up in the trees, and we're looking at things that maybe if we were going mushroom hunting and you see a lot of the really obvious things there, you're studying for your dissertation. Talk about that.
Yeah. So I'm studying a fungus called Parvidontia relampaga, and it causes relampago blight on lots of different types of trees and plants here in Florida. And it was described by a previous grad student in Matt's lab a few years ago. It looks like white lightning-like cords on the branches and leaves of these plants, and it'll colonize the leaves and kill them over time. And so if you're on the ground looking for mushrooms, it's very likely that you'll miss it because it's in the canopy and the branches above you.
How widespread is it?
When I first started this research, we only knew it from Central Florida, but now we have collections from across the entire state and a few in Alabama.
Gotcha.
Yeah.
And just within a certain like forest area, how likely are you to look up and see? Because I mean, it seems like you saw it quite a few places.
Yeah, I think it depends on the forest, but in Wakulla, it's like gangbusters. It's the craziest place I've seen. It's almost every other tree there, but in some other forests, it's a little bit more rare. And if it's more of like a scrub, sort of grassland sort of thing, you won't see it very often, but you still will see it on the big oaks and stuff.
So it's called a blight, which to me, as a gardener, that's a worrisome sounding thing. How worried do we need to be about blights?
Blights generally, I think it depends. I think with this blight, not very much. It doesn't outright kill any trees that at least from my observations. It does kill like the branches and some of the younger leaves, but it's not going to like destroy a forest like chestnut blight, for instance, or something like that. Yeah, so little on the worry scale.
Right. You've moved here from Cincinnati, correct?
Yes. Yeah.
So what brought you to Florida?
Matt did. I worked with another mycologist at Michigan State University, and they had been long-term collaborators. And we went on a collection trip together in Kentucky one year, and we did just what we did today, but we're looking for specific types of fungi in the order Pisizales. And so I got to meet Matt, and we chatted. And one of the Courtnarius we found, I mentioned this morning that Matt taught me like a cool trick about them. And that really stuck with me, and we had worked together for a long time. So when I was looking for to go to graduate school, he invited me to come to his lab, and here I am. So, yeah.
Basically, we got lucky.
Yeah.
That he chose to come to our lab. Yeah.
Does Florida have more diversity of is it like other types of things where hotter, wetter place maybe you have more diversity of certain types of things, or is that true for fungi?
My gut is to say that it's true, but since I've been here, we've been in such a bad drought that it's been hard to go out and find fungi. So this is one of the few times where I've gone on a collection trip not looking just for relampago blight, and this is one of the few times where the weather's been so nice to where we're seeing mushrooms everywhere. And so, at least from today, it definitely has a lot more different types of fungi than we see in the Midwest.
Right. And so you say, Matt taught you a trick for identifying mushrooms. So let's talk about this aspect of it. I'm sure, and you also work with the extension. I think you get a lot of like people coming to you and they want to know about a lot of things. So when there's a possibility of there being 3.5, and obviously they're not all right here in Florida, but when there's such a diversity, especially of things we don't even know that exist yet, how do you go about identifying mushrooms?
Well, it can be very challenging. I would say when you move to a new state that you have not lived in, and then you're in charge of that, it is a very fast learning curve. I've been here for 15 years now, so I feel like I'm getting pretty comfortable with knowing the most common things. So there are some disturbance-adapted fungi, and they show up in your lawn. There's definitely some that they go where we go, and those are more common. They're easier to identify because they're so common. But when you do have this overwhelming amount of diversity, sometimes you cannot tell from just a photo what it is. So sometimes you have to get a specimen and look under the microscope. Occasionally, I just need a lot more photos, and I can help. Part of that job is to work for the poison control. So when it's something that might have poisoned someone, then you want to get the information as fast as you can, and you need lots of photos, and sometimes try to get the specimen as quick as you can. But yeah, it can be very challenging, and especially when people send you one blurry photo from the top of a mushroom, you might not be able to get it perfect the first time.
Yeah. Let's talk about that, though. Photos. I mean, I do a lot with iNaturalist, too, and so it's true of anything. But even if you're just putting it on iNaturalist or if you're sending it to a researcher, what makes a good set of mushroom photos?
That's a good question. Do you want to answer that one?
Yeah,
Alassane?
sure. It's having a lot of photos. Distinguishing features are different for different types of mushrooms, but generally you want to have a photo of the top, the surface. If you're taking a picture of like a characteristic mushroom, you want the cap, and then you want the pores or the gills or whatever it has, because that can separate a lot of mushrooms. And then you also want a picture of what it's actually growing on, because that's very informative. If it's growing on wood or on the soil, or near water, or near the road, or in a lawn, that can all be very informative. Because as we saw, there's a lot of fungi that look very similar, that do very different things, and that can be a hint to sort of differentiate some.
Yeah, and then the other thing I'll add to that is when people are harvesting them, sometimes they'll see, if you teach them, they can learn to see these cues. Like sometimes you can see the spore color on that mushroom itself. Like there's a place where the spores are being deposited. It's kind of sticking to the stipe. You're like, oh, this one has brown spores, not white spores. That can be really important because sometimes you have two things. One has brown spores and one has white spores, but otherwise in the forest, they look really similar.
Yeah.
So, and then the other thing is sometimes when you're harvesting it, you will touch it and it will change color a little bit, or it will release some latex like we saw in the field today. So those things can also be really helpful. And you can learn how to photograph those if you're careful, but if you don't know that, you just take a picture of the top of the mushroom. We're having a hard time with that. Yeah, it's red. It's a red cap thing. There's only 1,000 species that look like that. Yeah.
And then sometimes too, when I've tried to figure out what some of them are too, I guess sometimes people don't realize how much they change from when they first come up, and when they start degrading, like they might look a whole lot different. Like you'd think it's a different mushroom. So talk about that part of it.
Yeah, definitely. They're aging, right? They're making it's just like a piece of fruit. You can think about it like a piece of fruit because that's what a mushroom is. It's the fruiting structure. It's making the spores. And if you have a piece of fruit, the day that an apricot comes out on a tree is really different than a week later what it's going to look and smell like. Yeah. And that can be true for mushrooms too.
Yeah, I asked you this question this morning, and you gave a good answer. So when you're how much does harvesting mushrooms affect the actual fungus? And then maybe this is a science podcast, and people listening might not even fully understand that the mushroom itself isn't the full organism.
Right.
Let's talk about that.
Yeah. So let's imagine a rotting piece of wood that has mushrooms coming out of it. A vast majority of the fungus itself will be the thin thread-like cells that are the body of the fungus, and those are the hyphae, and they make up the mycelium, and that's most of the body of the fungus. And so throughout the whole year, it'll live in that wood and decompose that wood, and then when it's wet enough or humid enough, it'll fruit and make that mushroom. And so by picking that mushroom, you're not killing the fungus that's in the wood. You are, I guess, maybe you're doing a little harm because it took a lot of energy and time to make that mushroom, but it's still able to disperse its spores, and it's still able to make more mushrooms. And oftentimes when you do see, there's a lot of mushrooms that don't make just one mushroom at a time. They'll make multiple. And so if you pick some and leave some, I think you're not doing very much harm at all.
Right.
Yeah.
And so I guess that also leads to, we're talking about, I want to say that before we get into this other part of ID, which is some people, if they get really into it, even as a hobbyist, and they're just enthusiastic, they want to learn, talk about making spore prints.
Spore prints are very important, especially when you're learning how to identify something. The spore color is a really critical thing. So certain groups of fungi have very distinct spore colors. It'll be, white or a purplish black or a reddish brown or a yellowy. And so you basically take the mushroom, wherever the spore bearing surface is, you're going to put it in contact with a piece of aluminum foil or a piece of paper, and then cover it up so it'll stay moist in there. You need that moisture in order for the fungus to be able to disperse the spores, and they'll be shooting off the little cells that make them, called most of the ones that we are talking about for that are basidiospores, and they'll shoot them out and then they fall down onto the paper. And when you get enough of them, you can see the color. And that is really quite critical, especially if you're just learning, to make sure you know for sure what the fungus is.
Yeah, it's kind of surprising to me because you think that the gill color would correspond to the spore color, but that's not always true, right?
It is not always true. It's often true, and you get better over time at distinguishing these things. But especially when I first started, I made a spore print of every single mushroom that I collected, and that really helps over time.
So we were out collecting today. So talk about the process of collecting and sort of how you preserve and how you study the mushrooms that you collect.
Well, you go outside and you get on your hands and knees and you look around. Most of the fungi we saw today are not that big, so you really have to slow down when you're walking and look in the leaf litter or on the sort of pieces of logs and whatnot. And when we collect them, especially for like things that look like amanitas, we want to dig down and make sure we get all of the structures of the fungus. So it's important you don't just like yank it out of the ground because you could lose some important features. And then we have like tackle boxes and we put all our fungi in there and they're nice and organized. And so we do that for the whole morning. And then when we get back to the lab, we'll put them in a food dehydrator and dehydrate them. And that way they're preserved for future study. But if we're going to work with them that same day, we can use them fresh or keep them in the fridge for a few days, depending on the species.
Yeah.
Yeah.
That's why you're doing different types of tests on different things to see, just cutting it or adding, so talk about some of these different little things you did to get the mushroom to sort of tell you a little bit about itself.
Mm-hmm. Yeah, well, today we looked at one really, what I thought is a really cool one that when you cut it, it's the flesh inside of the mushroom is maybe tan or gray, a little gray, and then it becomes reddish and then black over a period of maybe 4 or 5 minutes. I think that's a great one. It's an awesome one to show to kids or to students so they can see, okay, this is what some of these staining reactions look like. Not everyone is that exciting, but the chemistry that's going on is not always known, but it's like there's some chemistry that makes it specific to a certain mushroom that can be quite helpful. Some of them, you just handle them and they change color. And that can also be quite helpful for learning how to identify things.
Mm-hmm.
Yeah.
We also use things like potassium hydroxide, and some fungi will change color when you introduce that to them. They'll turn brown or something like that. But that's also informative.
Today we had one of those, and it turned red, and that's very distinctive. Yeah. Otherwise, kind of a little yellow mushroom. It would have been hard to identify, but that can be quite distinctive.
Yeah.
So mushrooms grow out of a lot of, decomposing plant matter. And so talk about, like, people see that in their lawns, and people get really concerned. So talk about sort of when we see mushrooms in our lawn, how should, and they ask you if there's good ones and bad ones. I mean, what do you tell them when people start asking you about this kind of stuff?
Well, so there are some mushrooms people get concerned about. I mean, there are some that are, varying levels of poisonous to people or toxic to people if you consume them. One thing I like to make sure people know is that all of them are non-toxic to touch. In North America, you can handle any mushroom that you get and touch it. You can smell it, get it up close to you, and that's fine. Any of the toxins from fungi are only problematic when you consume them. And I only recommend people consume them when they really know 100% what it is. Because as the poison control person in the state of Florida, I don't really want to hear about you and being not careful.
Other than toxicity, are they good or bad just for your lawn health?
Yeah. Well, so I think part of the problem is that fungi get a bad rap because there are some that are plant pathogens. I mean, we're in the Department of Plant Pathology, and a lot of people are studying fungi, and those fungi are very problematic for crops. So that is a legitimate problem. But the mushrooms that are showing up in your lawn, most of them are just decaying when you cut the grass, they're decaying the extra grass that you don't pick up off your lawn, and they're not really bothering you. And so I would say it's okay for them to be there. People get concerned about it. Sometimes they want to try to get rid of them, but really, I guess I see it as this is part of the natural process. If you have a more diverse lawn or a more diverse yard where you leave out pieces of wood, you allow there to be native plants, you'll have a pretty diverse group of fungi. And if that's happening, that means you are, cultivating diversity in your yard. So I try to encourage people to think about it like that.
Also, I can't imagine if I were to think of getting rid of a fungus, if its mycelium is all throughout the soil, you'd have to just get rid of all your soil to get rid of the fungus,
Right.
Yeah.
Almost impossible.
Yeah.
Right. You talked about mushrooms that, would colonize a disturbed area. So let's talk about that a little bit. So, disturbed soils. And so a lot of soils in our yard don't have, this mycelium or the mycorrhizal fungi in them. Talk about sort of mushrooms as an indicator of soil health and...
Yeah, fungal diversity is in a forest, for example, where we were today. If you take a handful of soil, there's hundreds of species potentially in that one handful of soil.
Yeah.
So it's normal. It's natural. It's what's supposed to be happening in soil to have all those organisms there. When we disturb the soil, like for example, when they do strip mining and they take off the top of the mountain, basically, they're taking away all of that diversity, and then they're also leaving very little organic matter in the soil. And so to get that back, there's kind of a succession. A lot of times what has worked for when people are trying to sort of revegetate is you bring in a hardy plant that has mycorrhizal fungi like a pine. You bring in some of those mycorrhizal fungi and they can often get started. And then over time, you'll see, new plants coming in, new fungi coming in, and this buildup of soil carbon again. But it can take a long time for that process to happen if you've gotten rid of everything.
So a lot of it starts with trees then. Do a lot of tree species already come with associated fungi like that?
Yeah, like for example, pines, if you try to, so there was an experiment that people did where they tried to plant pines and have pine forestry in the southern hemisphere where pines are not native. So South Africa, the tip of southern South America. And lo and behold, the pines did not grow when you brought seeds. But if you brought seeds in just a handful of soil, you got mycorrhizal fungi. And when you bring that soil, you get the mycorrhizal fungi and they grow great and it's no problem. In fact, they become invasive in some places, pines in the southern hemisphere, which is another issue, but they're very successful in pine forestry once you have the mycorrhizal fungi.
Going down a little bit of a rabbit hole here, just because, I write a lot about native plant gardening and all that. And so if you're introducing native trees into an area where they belong, and maybe native ground cover plants, are they likely to work together better on that level?
I would say yes, they're much more likely to be able to contact their symbiotic fungi. I will point out that they often are also contacting their pathogens. And people are disturbed by that, but within the natural context of the plants being where they're supposed to be, you will have pathogens. You'll have insects that eat them, and you'll have fungi that'll eat them. And that's the normal process.
Yeah. I think if you look at a majority of plants, they're unhappy. They're sick. If you look at the leaves, there's some leaf spots or decomposition happening. I think a vast majority of plants have some sort of disease going on or pest, Yeah, even if they look very healthy.
And it's part of the way it's supposed to be. There's a lot of carbon there, and the fungi want to get it.
Yeah.
Yeah, and so do the insects. It's sort of part of the plant evolution too, right?
Yeah,
that's how they build the resilience.
Yeah.
So I want to shift now to we were looking at a lot of logs that had fallen, and we weren't even seeing like mushrooms in them, or even white streaks of anything always. But like, talk about some of the different things that we were seeing in like that brown fungus that was breaking down the logs.
Oh yeah, you want to talk about the types of decay?
Yeah.
Oh yeah. So there's two sort of broad classes of wood decay that we see very often. And the first is brown rot. And when you see that, the wood or the log looks brown. And it's got this brownish reddish color. And when you sort of break it apart, it breaks it apart into like cubes. And that's because the fungi that are decomposing that plant are only decomposing cellulose, which is very easy to decompose. And so all the lignin is left in the plant. And white rot is caused by fungi that can decompose the lignin and cellulose. And when you look at that wood, it's white, and it's sort of spongy, and you can take it and you can pull apart almost like shredded chicken. And so there's like very little plant material left in those types of rotted logs and whatnot. Yeah.
Yeah. Can we put that in the context of when a tree dies in the forest and sort of this cycle of what it, its journey to soil, I guess.
Mm-hmm.
And so talk about fungi and sort of the, this role they play in the forest, which is what the forest needs, right?
Yeah, they're critical because otherwise everything would be locked up in all this old organic material from plants. And instead what's happening, right, is those fungi are breaking down that material. And you can see it really well. Like when we were looking at these brown rotted logs, They hold a lot of moisture. You get fruiting of all these other fungi later in the succession. But then we saw, for example, this seedling coming out of right at the edge of one of these. So you can see this soil that's slowly being created. The fungi are the ones that are sort of running the carbon cycling. And then at the same time, the other part about why the mycorrhizal fungi are so important, well, we saw a lot of mycorrhizal fungi today that were fruiting on the forest floor. And when you see them, you can't necessarily look at the fungus and know, okay, that's a saprotrophic fungus or that's a mycorrhizal fungus. But once you get experience and you know sort of some of these different groups, you see that some of the ones are mycorrhizal. What does that mean? It means they're basically using their hyphae to go into that organic material. They're extracting the organic nitrogen. The organic nitrogen is not available to the plants. So plants are pretty good at getting inorganic nitrogen, which is why we give them, fertilizer in our garden. But that organic nitrogen is locked up in a way that the plants don't have access. So those fungi are, growing into that organic material with their hyphae, extracting that nitrogen and trading it to the plant in exchange for sugars. And sort of that process is very important to the carbon cycling as well in the forest.
Yeah, and I also will say that they're important in starting the decomposition process because we saw a lot of logs that were rotting, and then they also had insect holes and like evidence of worms and all kinds of things going in there, and also speeding along the decomposition. And so that wouldn't be available without the fungi starting to decompose the difficult plant matter.
Right. That's the first thing that starts like breaking down the log is the fungi, and then once it's soft enough, and then insects come and they build their burrows in there, and everything from bees and beetles and all kinds of fun stuff. I want to get back to mycorrhizal fungi. So you're talking about, there's this relationship between the fungi and the plant, but also it connects plants to each other to some degree.
It does. Well, so people talk a lot about mycorrhizal networks, and it's a cool idea. And I don't want to say it doesn't happen, but I think it's important because I think about things from the fungi perspective. I think about it in the terms of the fungus is going to do that, but it's only going to do it when it's good for the fungus.
Right.
And sometimes people think about fungi as like in that context of mycorrhizal networks, they think about the fungi as sort of these passive tubes that are just transporting things between plants. And they're happy to be transported between plants if they get something out of it. And if they don't, and the second they don't, they'll be happy to eat the plants. So I guess that I think it's important to think about them as autonomous actors in this context. They are happy, and many of them, they are happy, quote unquote, happy to act as symbionts, but that means they are getting something out of it too.
Right.
And that's why it works. And as soon as it doesn't, they won't do it. Right?
Yeah. I remember hearing a lot about, when I first started hearing about it a few years ago, the wood wide web, and it's like, these trees are all communicating with each other, and they're sharing water, and it's I guess anthropomorphize it a little bit, and
well, it's good though in the sense that it gets people to think about what is going on under the soil with the forest, and it's not something that's intuitive and obvious to people. Even tree roots, you don't see them unless the tree tips over. You don't see them, and you don't realize how extensive they are. And for these fungal networks, but also fungi in general, I mean, they're everywhere in the forest, and we're walking on them all the time, but we don't think about them, or most people. If you're obsessed with fungi, like some people, you're thinking about it all the time, but the average person isn't really thinking about them until it's a mushroom on their lawn that they're unhappy about.
Yeah.
So.
Yeah. And so I guess I see a lot more and more all the time about sort of what lives in the soil and these. So how much do we understand about that aspect of fungi?
Alassane, you want to give that one a try?
Close to zero. It's very little. So like Matt mentioned, there's so many fungal species that we just haven't found or described formally. And when we were walking today, we talked a lot about how a lot of fungi or a lot of mushrooms look similar to each other but are completely different. And that's like one part of the hidden diversity of fungi. But soil is this big part where there's so many fungi that don't make mushrooms, that don't have sexual parts of their life cycle that we know of, or don't have really distinguishing characteristics. And so we know by sequencing the DNA in soil that there's a lot of different things. But it's very hard to go in and like look at how they're different or what they're doing. And I'll say too, like a lot of the species that we've described because we're able to culture them and grow them up in a lab and study them further. Whereas these soil fungi, a lot of them we can't culture in lab conditions, or even mycorrhizal fungi that we know we have a good idea what they're doing. They make mushrooms, but we can't culture them in lab conditions because they don't have the correct nutrients or the conditions.
Yeah, I guess I'll strike an optimistic note, which is to say there's a lot we don't know, but what we know has really exploded in the last 25 years, I would say. Like when I took mycology as a student, as an undergraduate student, versus when Alassane took it is really different in terms of the molecular data, in particular DNA data, that's really revolutionized our understanding of fungi. So like those patterns of convergent evolution, some of them we thought maybe, but now we have really good evidence of how much this convergent evolution has happened. The idea of diversity, how much diversity of, in terms of species diversity there were some ideas, but the molecular data has really given us a lot more to work with. So we can, for example, do things like make species-specific primers or probes and follow a particular fungus in a particular environment. And that really wasn't possible 20 years ago or 25 years ago. And now we can do a lot of things that we couldn't do before. And it has really changed a lot of our basic understanding of fungal biology in a very serious, very revolutionary differences in how we understand them. So that part is pretty cool.
Yeah, I definitely take it for granted. And when you read older papers, it's like, oh, why didn't they sequence everything?
Yeah, right. Because for a lot of people, their dissertation was one sequence of one fungus in the beginning because it was so hard to do. And now we have the ability to just generate so much information, which is exciting. It's a really cool time to be a fungal biologist for that.
Yeah, so you described mycorrhizal fungi, and you also used the word saprotrophic. So what does that mean?
Alassane, take it.
Saprotrophic fungi, they are decomposers. They decompose wood or, like Matt mentioned, like grass. So I guess how many groups would you say? There's like mycorrhizal fungi, so those are the ones that associate with plants. There's saprobes that decompose things. There's plant pathogens. There's human pathogens. There's pathogens of other fungi. There's pathogens of insects.
Yeah.
There's a lot of different ways to get energy that fungi have and have evolved. But I think a vast majority of the ones we collected today are either saprotrophic or mycorrhizal.
Gotcha.
Yeah, when you walk around in the forest and you're randomly choosing mushrooms off the ground especially, it's probably 50-50 that you'll find a mycorrhizal fungus, depending on the forest, but often mycorrhizal fungi and saprotrophs are kind of the dominant thing. One other thing that I think, it's a little bit of a tangent, but I think it's in talking about how things have changed. So with genomics, one thing that's really cool is that we can use genomics to interrogate some of the questions that people have had for a long time. So you may have these fungi that you can't grow them in the lab, but you don't really know what they do. So now I would say we're able to sequence genomes and reconstruct the genomes and then take some guesses about what they might actually be doing in the environment. And there's a lot of exciting things that are coming out of that. One thing is that a lot of these fungi are doing probably multiple things. So they might be doing one thing one part of the time, and then in another part of their life cycle doing another thing. So there's like these big changes in trying to understand the biology of them because we have all these new tools to really dig in, I guess.
Yeah. Do you have like an example of, because like ectomycorrhizal fungi, you were mentioning this morning about how a lot of ectomycorrhizal fungi evolved from saprotrophic fungi. And so when we look at their genomes, a lot of ectomycorrhizal fungi will not have as many wood-degrading enzymes as their saprotrophic relatives would have.
Yeah.
And so that's how we use the genomes to look and see, can we predict whether or not this is ectomycorrhizal or saprotrophic based on the proteins that it's able to produce.
Yeah, that's a perfect example of some of the things that we can do that we couldn't do. The other thing is we have more examples of these things that have been shown experimentally to do more than one thing. So we worked on one in our lab. It's this genus that's a bolete. We saw a lot of boletes today, and most of them are mycorrhizal, but we worked on this one really strange one, and that one you can grow it in the lab, so it's very convenient. You can grow it in the lab, and you can show that it does wood decay under lab conditions. You can make it decay wood, but then you put it in plates with other fungi and it actually attacks those other fungi. So this is one of these weirdo ones that it's a mycoparasite and it's a saprotroph. And I think in our heads, we like to have these categories like it's this thing or it's that thing because that makes sense to humans, but they don't necessarily, they don't believe in the same confines.
Yeah.
They want to do whatever they're going to do to make a living. So that makes it different.
Yeah.
Yeah. And there's some other categories too. Coprophilous fungi, is that?
Uh-huh.
We did something with archaeologists, and so they're able to look at it since they feed on herbivore dung.
Yeah.
They were able to tell when mastodons went extinct in this area, for instance.
Yeah.
Let's talk about those a little bit. And I guess there's one that I guess people probably ask you about a lot.
So there are some famous dung fungi. So yeah, there are some specialized fungi that are only on particular types of substrates, and one is dung fungi. And so mycologists really love dung. They love herbivore dung because you find all kinds of very specific fungi. I would say in the public interest, there's one particular dung fungus that you can find in Florida that is psychedelic. So I get a lot of questions about that because it's one of the ones that people consume. It is not legal. So I'll say it's not legal to have that fungus, but I still get a lot of questions related to it.
And does it have lookalikes that are maybe kind of toxic?
Well, so with any of the psychedelic fungi, they can be really hard to identify. So just like anything that you're going to consume, you want to be 100% sure what it is. So I will say there are some for the not very experienced, there are definitely some wood decay fungi that are on the forest floor that look similar to some of the psychedelic fungi, and there have been lethal poisoning events from that in the U.S. before. So it's not common, but you still want to really be 100% sure when you're consuming anything. I think it's a good rule of thumb.
Right. And I guess that leads into something, I guess I'm part of fungus Facebook groups, and people try to identify things and share things. And for the most part, it's about, look at these cool, but every once in a while, people sneak in with that kind of thing. And also questions about edibility. So talk about sort of the good and the bad of that these groups exist to get people excited about that, but then also there's all this other aspect to it where, there's some potentially like you're asking people online that you don't know if you should eat this or not, or that kind of thing.
Yeah. I think it's hard, and I think in Matt's fungal biology class, he talks about this a lot, to where you have to just choose a fungus that you really want to eat or you're interested in, and make sure you know all of the lookalikes for that fungus, and you make sure that you know all the distinguishing characteristics for that one fungus before you ever are sure about eating it. And then also ask a specialist like himself, or I think getting multiple lines of evidence that it's the correct fungus would be the right thing. I'm a mycologist, and I don't eat any amanitas or blights or anything, and I can identify them, but I'm wary. So yeah, I think, for instance, like the chanterelles, there's lookalikes that we have in Florida, and they might look very similar when you glance at them, but when you know what you're actually looking for, they look quite distinct.
Yeah, I encourage people to identify a fungus multiple times until they really feel comfortable with it. They know the environment it's found in. They are comfortable with things that look kind of like that. That's a good, you wouldn't go out and eat berries if you weren't 100% sure of what you're going to be consuming. So I think it's kind of the same standard. People don't eat plants that they don't know what they are, and it's a good way to not be in the hospital and hurt yourself. And it's the same with fungi. I do think with training you can learn, but the problem is sometimes people want the quick answer. They want you to answer for them, and that means they're probably not ready to be foraging for their own food like that.
There are a few that are like very easy. I haven't seen chicken of the woods here in Florida, but is it around?
Oh yeah,
yeah.
So, neighborhood.
Like there's.
It's very distinct.
It's very distinct. I think it's hard to mess that one up, and it's very common in the U.S. But the things that look like what are the lookalikes for the death cap in California that people accidentally collect all the time?
To be honest, there's not very many. Well, I guess there are some amanitas that are edible. I never encourage people who are even experienced people can make mistakes, and so you have to know what are the toxic things, and that's important to get to know. And so in Amanita, for example, there are some edibles in the genus Amanita, but many of the largest deadly poisonous mushrooms in the U.S. are Amanitas. So I don't encourage people to eat from that group.
Yeah.
Yeah. Speaking of, so one of your research interests is truffles?
It is.
Are all truffles edible? Or
Well, I study truffles in the broadest sense. So some people would say truffles are only this one genus tuber.
Okay.
That's kind of the famous truffles are all in the almost all in the genus tuber. And so some people have sort of a strict sense that truffles are only the genus tuber. I am interested in truffles in the broad sense. And this is another convergent evolution thing that's shown up all over different groups of fungi have evolved this sort of go below ground strategy. Usually they give off some sort of odor or visual cue and an animal digs them up and eats them. So I will say they're edible for someone in the forests. But there are a lot of them we don't know if they're edible or not. Some of them have really, really strong odors. Some of them you wouldn't want to eat. They smell a little stinky. So I don't generally eat truffles unless they are like known in the genus tuber or a couple of other genera. We've worked a lot on this genus Leucangium, and there's a species that is called Leucangium cascadiense in the Pacific Northwest. And so there are these ones that are known edibles they're very tasty in my opinion. But yeah, I'm really interested in this convergent evolution and the fact that there's so many different fungi that do this below ground fruiting strategy. I'm curious in why that happens and where it happens and how much diversity there is, things like that. A lot of those are also mycorrhizal fungi, and I'm really interested in mycorrhizal fungi too.
So just define convergent evolution.
Convergent evolution. So it's basically where you have the same feature evolving over and over again in different groups. So you can think of birds and bats as a good example of convergent evolution, right? Two unrelated groups of organisms that have evolved flight, but they have some similar morphological structures. And you would say, those are organisms that fly. So those are examples of convergent evolution. We have lots of those in fungi. Which makes it challenging, but also pretty exciting.
Yeah, you're talking about lots of different groups of related things. So before this, I guess I used to think, mycorrhizal fungi were just a type genetically of. This is more of an evolutionary behavior, ecological behavior.
There's multiple kinds of mycorrhizae too, so that makes it a little bit confusing. But I would say mycorrhizal fungi in general is a broad term of these fungi that associate with plants. The ones that are what we call ectomycorrhizal, those have evolved lots of times, more than 70 times. Whereas things that you'll hear of as called arbuscular mycorrhizal fungi, so there's evidence to suggest that those were here for 400 million years, and that when plants were first moving onto land, when there was basically no soil, that those fungi were associated with those primitive plants, and that they were probably really critical in that process of moving from aquatic environments to land environments, especially for the development of soil. And so those arbuscular mycorrhizal fungi, we think they actually only evolved one time, and they're very diverse and all over the planet with plants. So they're kind of these two different groups. There's more, but those are the majority of the mycorrhizal fungi.
Yeah. So let's talk a little bit about some of the things we found today, and I have you on camera too, so I'll edit some of that in. But we have some right here.
Yeah.
What are some fun things to? Yeah. What are some of your favorites here?
Alassane, you want
one of my favorites. I've never seen this before I moved. I had never seen this before I moved to Florida. Is these xylaria growing out of the magnolia cones? Should I describe it as if we're in the field? I don't know.
Well, let's talk. So there's a video version for this, obviously.
Okay.
Let's just describe for people listening at home who are only listening on a podcast or on the radio.
Okay.
What does this look like?
Okay, so if you imagine the magnolia flower, this sort of my plant biology is pretty bad. The cone part of the magnolia.
Yeah.
Once it falls onto the ground, it'll sort of turn black, and then these black sort of finger-like structures will grow out of the cone. And it's this fungus called Xylaria magnoliae, and we believe it lives inside of the magnolia leaves and cones. before it falls onto the ground, and then once it falls onto the ground, it'll consume it and make these fruiting structures. And that's one of my favorites that I've found in Florida because it's pretty alien looking, and I hadn't seen it before I moved here.
Right.
Yeah.
And there's another one that's a different one that comes out of magnolia cones too that has the little caps on it.
Yes, there are multiple things that will fruit on magnolia cones. I think you're probably talking about a Bayospora. There's one that is a Bayospora that seems to be sort of specific, I believe. Yeah, so there are some plant substrates that will have very specific fungi fruiting only out of certain parts, but
Yeah.
that's not necessarily, those are a little exceptional. This one is very exceptional just because it's on this very distinct plant part, and
Yeah.
it only seems to fruit on that,
Yeah.
Yeah.
which is pretty cool. I guess I really like these black chanterelles quite a lot. The Craterellus, they're just really pretty. I don't know. They have this kind of sheen to them. They have a really interesting shape to them, like the horn of plenty or whatever you want to call it. I just think they're really pretty. They smell really good. So I don't know. That was one of my favorite. That's one of my favorites that we don't see all over Florida. So you need really nice, wet hardwood forest. Where we were today was great for that. So if I see that, I know I'm in a good place for fungi because it's not common all over Florida.
Another treat I saw was the earth stars. I hadn't seen that really ever, unless from photos. And so that was really exciting. And so it looks like a ball inside of another ball that makes this sort of star structure that holds the fungus. And it works almost like a puffball. So it has a little pore on it the spores will shoot out of. And they've always been really interesting to me. And it was good. We saw quite a few today.
Yeah. Looks almost like a star or like a sun or a flower almost.
Yeah,
It
yeah.
almost opens up like petals. It looks
Yeah,
like a little bit.
exactly. Yeah.
Yeah.
One thing that you can see actually, because I think it's informative because we talked about the aging of the fruiting bodies. So a lot of these really small ones, you can see we kind of have them clustered here together. You can see already that they're starting to look a little bit worse for the wear over here. So these small, delicate fungi, they're already getting a little bit tough to look at. I mean, they don't have the same color. They probably don't have the same odors. They're reduced in size. And this is one of the issues, right? These really small things are hard to collect, and you have to get them back to the lab and look at them before they're really drying down to nothing. Luckily, a lot of the morphological characters that are microscopic will be preserved.
Yeah.
So we could go back to the lab and still get information from them.
And that's another reason it's really important to take more photos than you think you should when you're in the field, because a lot of those features can sort of disappear. The colors may fade, or the shapes will change, or the smells will go away. So taking as many notes as possible and getting as many angles as possible is really helpful.
So smell's a big part of it, huh?
I mean, it definitely can be. There are some that have very distinctive odors, and that can be really important in determining, between two different species. So we have one in here. Let's find it. There's one in here that has this really nice odor. I really like this one. This is a Lactaria species that we smelled when we were out there. And you can see that it's like, not very exciting. But when you take a sniff, this one's like maple syrup. I really like that one. Those are called the candy caps. So I really like those. They have a great name too.
Yeah.
Yeah. So talk about you teach, obviously you teach graduate students, but you teach undergrads as well?
Yes, I have a 20% teaching appointment. So I do extension, research, and teaching. But for my teaching, my main teaching responsibility is fungal biology. So I teach fungal biology every fall. And it is a mixed class. So it has graduate students. It's mostly PhD students in our department in plant pathology, but also we get people from food science, from entomology, from other parts of campus. And then upper division biology students take it. And it's great. I really enjoy it partly because there is so much still to learn about fungi that I learn new things every year. So that makes it exciting for me. It's not just rote memorization. It's new to me every time. So that's pretty exciting.
Yeah. And a big portion of the class is what we did today. There's a big collection project where you have to collect fungi across the semester and try to identify them by yourself. And so it's a really good hands-on way to understand the convergent evolution, or understand what traits are common in what types of fungi and things like that.
Is there anything I didn't cover that I should have covered? I mean, there's so much we could talk about, but as
I
far
was
as,
gonna say we
as
could
far
go
as
on all
people-
day. Yeah.
As far as people, people who go to parks and see mushrooms, it's just as far as it would enhance people's enjoyment or understanding of what they see when they see mushrooms.
I guess I would just say I find that many people are scared of fungi in part because there are these deadly poisonous species that if you eat them, it's not going to go well. And I can appreciate that. But I find that when I meet people and I start talking to them about fungi and I tell them like, you can handle any of these fungi, it's going to be fine. You should smell them. You should pick them up and see them. They feel a little more free to like, okay, it's not going to hurt me. I think fungi have a bad reputation a little bit. So I guess I would say, I think we did this with the podcast in general, and with our time in the forest, but I just like to encourage people to like take the time to look at them. Don't be afraid of them. And you might start to really think they're cool and want to look at them more. That's what I found anyways. Yeah. And I found that many people, once they're not scared, they can do that.
Yeah. And I think a good way to get involved is just taking pictures of stuff and putting it on iNaturalist. I think many of my ecologists I've spoken to have found a collection of some fungus that they didn't expect to be in one part of the country or another by using iNaturalist, and they're able to talk to the person who collected it and try to find out where that was. And that's very helpful. When we were first doing this research on Relampago, and again, we thought it was only Central Florida, we got someone posting a picture on iNaturalist of it in, I think, Fort Myers, and that was really exciting to us because we didn't know where to go in South Florida and we had no idea that it would even be there. And so even if it looks very common or maybe boring, I think it will be helpful to people in the future.
Yeah, that's a great example where, if you take good pictures like we've talked about from multiple angles, even on your phone, phones are really good these days with the photos you can take. That sometimes leads to really exciting discoveries in fungal biology. So that's a great example that we encourage that.
Yeah, and it's good to hear that you use the iNaturalist data. So I've talked to other researchers too, and it's good to know that it is being used. And so when people, it's a thing that can teach as an individual, it can teach you things if you use it the right way, but also is helping research.
Yeah. Absolutely, because it's, fungi are such a sort of understudied group. It can really help a lot.
Yeah. And then I guess one final question is, as mycologists, are there mushrooms that you travel to see?
Oh, that's a good question.
I guess I've been traveling to collect the Relampago blight because I have to. But I don't know if there's any that I would like go out of my way to see. I think I'm going to say no, but because everywhere I've been, there's always been something new that I haven't seen before, or something that I have only seen in a textbook or something. So, we don't have to go to like Bali to find a rare fungus. You can go in your back forest or whatever.
And also when you're looking for it, I'm guessing you just see a lot of things in general that you've never seen just because you go to see one thing and then you're looking around and like, oh,
exactly.
This is cool.
Yeah.
A lot of the time, the coolest fungus I find, I see something kind of big and I'm like, oh, and then I'm down on the ground. So I see, oh, wait.
Yeah.
And that happened today, actually, when you kind of were like, oh, we stepped right over this one.
I know, I was on the way to you because you were seeing something and then I looked down, I'm like, it was the shape too. Because on soil and with all the dead, you could barely see that.
Yeah.
Mm-hmm.
Yeah. Are there any that you travel to see though?
Well, I would say, like Alassane, I know that I don't need to travel to see them. We've found hundreds of species in the University of Florida Reserve that's right on campus. So I don't have to go anywhere, but I sure like to go to places where there's some really different fungi. So I have done travel, especially for truffles. I guess I'll travel for truffles because I love them. And actually, Florida's not a super diverse truffle place, but there are some other places where you find them a lot more. So I will take an opportunity when I get it to travel to some of those places that have a lot of truffle fungi.
Yeah.
Great. Well, thank you, Matt Smith, Alassane Sow, for coming on to Coast to Canopy.
Thank you very much for having us.
Yeah, this has been great. Thank you very much, Rob.
If you were listening to the audio version, I know you wanted to see the mushrooms we were talking about. You can watch the video version of Coast to Canopy at wfsu.org/ecologyblog and on the WFSU Ecology YouTube channel. I've been seeing a lot of AI-generated images, articles, and videos about our natural North Florida. Some of those make use of images we took on our adventures, without our permission. We're going out to these places and we're talking to the people who best understand them. That's the job. And that job is made possible with support from viewers and listeners like you. WFSU is both an NPR and PBS station, and we can't serve our community without your support. Here's a way to support. For a $10 a month pledge, you can join the WFSU Seed Circle, and I'll give you a shout out on this podcast, like right now. Thank you to Thomas Beeson, Karen Berkeley, Margaret Bivens, David Blair, Beth Garraway, Carol Goodyear, Veronica Humphrey, Lisa Johnson, Chris McFarland, Carrie Maddox, Clara Mullins, Mary Pohl, Alina Pryor, Thomas and Helen Roth, Dylan Riles Hamilton, Kim Sash, and Donna Tharp. We've also had several smaller donations, which I really appreciate. Any amount helps. You can join the Seed Circle at wfsu.org/ecologyblog. This has been Coast to Canopy, an ecology podcast from WFSU Public Media. Today we had Taylor Cox running audio and Jason Acuna directing video. And I am your host and producer of all things ecology at WFSU, Rob Diaz de Villegas.